Here is our DNA double helix, parent model, and you can see that the strands run anti-parallel from one another. The top strand is from 5' to 3', while the bottom strand is from 3' to 5'. Notice that the licorice represents the backbone, which is composed of phosphate and the deoxyribose sugar, while the rungs of the DNA ladder are composed of nitrogen bases. You can see that A pairs with T, the yellow and the green, while G pairs with C, the purple and the red. The nitrogen bases are held together by weak hydrogen bonds that will soon be broken by helicase. Helicase is represented by the Swedish fish, and the job of helicase is to unwind and unzip the parental double helix simply by breaking the hydrogen bonds between the nitrogen bases. We call the point at which helicase starts to unwind and unzip the DNA the origin of replication. Now we have an enzyme called DNA polymerase III that is represented by the peach O. DNA polymerase adds complementary nucleotides to the parental strand in the 5' to 3' direction only. We call this strand the leading strand because it is synthesized continuously. The leading strand is represented by the red Twizzler. Again, take note that new nucleotides can only be added on the 3' side of the growing DNA strand. So we say that new nucleotides are always added from the 5' to 3' direction. Now here's where things get tricky, the synthesis of the lagging strand. Notice how the direction bases can only be added to the 3' end is away from the replication fork. We need another enzyme to help with this process. That enzyme is called primase and is represented by a pink gummy lifesaver. Primase lays down a small section of RNA nucleotides in fragments called primers on the parent strand. The RNA primers are represented by yellow Twizzler pieces. The RNA primers act as a starting point for DNA polymerase III on the lagging strand. Once the RNA primers are set into place, DNA polymerase III comes in and adds complementary nucleotides to the 3' end of each fragment. These fragments are called Okazaki fragments and are named after the Japanese scientist that discovered them. Notice that once the DNA polymerase III completes synthesis of the fragment it is currently working on and reaches the primer ahead of it, it jumps back to the primer located behind it to continue adding nucleotides in the 5' to 3' direction. Once all of the nucleotides are set into place on the lagging strand, a new enzyme, DNA polymerase I, represented by the green lifesaver gummy, removes each RNA primer and replaces that area with DNA nucleotides. The last finishing touches are made by DNA ligase, represented by a red lifesaver gummy. Ligase means to bind or glue together. DNA ligase is an enzyme that will join the ends of each fragment together until the lagging strand is complete. Voila! We have just successfully completed DNA replication. You can see that we started with one parental DNA double helix, and now we have two identical double helices, half old and half new.